So today we're joined by Dr. Elaine Mardis. She's the co-executive director at the Institute for Genomic Medicine at Nationwide Children's Hospital, where she's a professor of pediatrics at Ohio State University College of Medicine. Elaine graduated with a PhD from the Department of Chemistry and Biochemistry at University of Oklahoma. And then she was a senior staff scientist at Biorad Laboratories before she joined Washington University School of Medicine. The Institute for Genomic Medicine at Nationwide has been with OSU, or she has been there since then. Elaine is an internationally recognized expert in cancer genomics. Oh, she's been there since 2016. I'm sorry, I missed that. And she has ongoing interest in the integrated characterization of cancer genomes, defining DNA-based somatic and germline interactions and RNA-based pathway and immune microenvironments, exciting stuff that lead to the cancer onset and progression. So she's going to talk about genomics-guided pediatric precision cancer medicine, which we all want to learn more about. And so with that, I'm going to turn it over to Elaine. Thank you for joining us today.
Yeah, absolutely. Thanks so much for the kind invitation to participate. And I hope what I've prepared today is of interest to those who are joining in. So as Jay mentioned, I'm going to talk a little bit about the work we've been doing here at Nationwide Children's Hospital in genomics-guided pediatric precision cancer medicine. And then I'll finish up with the newest efforts on the COG-related so-called MCI, the Molecular Characterization Initiative, that we've been really proud and pleased to be a part of for the final bit here. If I can just get this poll to stop showing up in my screen. So just a little bit of disclosure information. I have a few conflicts of interest that are listed here and should be pointed out, although they are not germane to the talk that I'll give today, but do reflect some of my outside activities with different corporate entities. So first, just a brief introduction to the IGM, who we are. We're obviously at Nationwide Children's Hospital here in beautiful, sunny Columbus, Ohio. And we are a bit of a unique entity. And so as you can see from the circles down below, these pictures of the hospital tower on the right and our institute building on the left. What we encompass within the Institute for Genomic Medicine is sort of the nexus of research as it transitions into clinical. And in particular, we have groups that focus on the development of technologies and the offering of genomic services to our investigators at Nationwide Children's as well as at Ohio State. We have focused research groups, including PIs who are part of our faculty within IGM that pursue genomics and molecular biology research. And these are exclusively in different areas that are pertinent to pediatric medicine that are also driven by genomics. And as I'll talk about today, we have multiple collaborative translational protocols. So these are active working groups within our different groups of providers in the hospital, where we are working towards the ultimate vision and goal of having genomics become a part of medicine in its practice and implementation. And of course, today, given the topic of focus, we'll talk mostly about the cancer translational protocol and what it has developed into over time and the technologies that we're using to drive it. Finally, and uniquely on the far left, the dark blue circle indicates that in addition to lots of research and translation going on here, we also oversee clinical, CAP/CLIA and New York State Department of Health licensed cyto and molecular genetics within our clinical laboratory that's part of IGM. Overarching all of these areas, especially with the advent of next generation sequencing and its use in clinical medicine, are a vast array of computational capabilities. And I'll try to give you a feel and flavor for those as I go along. So we all are focused here on pediatric cancer, as are you. It's the leading cause of death by disease in the developed world in children over 1 year of age. And we've obviously known for quite some time that cancer is a disease of the genome. This is one of my scientific heroes, Janet Rowley, a pathologist and a person who really embraced technology and its use in the practice of medicine in the early days of her career in the 1970s, really devising key chromosomal stains that would allow her to examine cancer chromosomes under the microscope, and in aggregate led to the early recognition of altered chromosomes as a key component of different types of leukemias. In 2021, I like to think that our next generation sequencing and computational capabilities are the molecular equivalent of what Janet was doing back then. And we're using this as our microscope to really evaluate children with cancer in our hospital. Last year, we used this combination to study 124 children with cancer, and we were able to return medically relevant information on over 95% of these cases. Even though this was a research protocol, in cases where this information was pertinent to the care of these children, we could reflex the specific alteration into our CAP-CLIA laboratory, perform an orthogonal clinical test, and in cases where this came back with a positive result, write this into the medical record for these children. I think the challenge, though, that we're talking about today in terms of the larger scale and scope of pediatric cancer is really survivorship. And this is a huge challenge across multiple areas, as illustrated by this nice review in JAMA Oncology a couple of years ago, wherein the evaluation was performed about patients diagnosed as children with cancer in the 70s, 80s, and 90s, looking at specific and all-cause mortality. This is just one figure from the paper showing the relationship of chemotherapy and radiotherapy to different components of mortality. And what we really understand is that although the long-term survival of these children has improved over time, as adult survivors of pediatric cancer, there remain significant risks of shortened lifespan and secondary cancer diagnoses, along with quality of life aspects that contribute to newer aspects of mortality that include suicidality. So our IGM studies are really committed long-term to changing this reality. And I'll talk a little bit about this today in the context of identifying targeted therapeutic vulnerabilities, really the norm in adult cancer medicine today, as Mike Berger and I wrote about a few years ago in Nature Reviews Clinical Oncology, where the combination of somatic and now germline variants actually can dramatically changed the therapeutic landscape for adult patients. And we really would like to imagine a day, hopefully not too far in the future, where this becomes the truth for our children with cancer as well. But of course, we have to do this recognizing the significant challenges that I've just listed a few here. You're probably all familiar with them. Firstly, that unlike adults, there are few mutations identified in these cancers. Really, this is the reason for the multiplex characterization I'll talk about. And treating our cancer patients with FDA-approved drugs that target specific mutated genes and the proteins that result from those genes is difficult because we poorly understand dosage, unknown adverse event profiles, and other aspects of, their interaction with small children that really, in many ways prohibits their use in a direct format or as off-label. Clinical trial enrollment until recently, where the ages of inclusion have dropped, have often excluded pediatric patients. And of course, there's now new legislation that encourages corresponding pediatric clinical trials for new agents and new therapies. But other areas where adults have benefited, like checkpoint blockade immunotherapies, are rarely used or even considered appropriate. So I think, we do understand the use of targeted therapies and immunotherapies may help to alleviate long-term consequences of aggressive chemo and radiotherapies, but there are a number of hurdles that we really have to get across before we can make this part of the reality. To address this, though, we thought it would be great to begin at least performing multiplex molecular profiling on our cancer patients. So in early 2018, we began this IgM cancer heme protocol, moving us from, as you can see from the graphic at the top, specimen intake from pathology assessment and recognizing the important diagnostic information coming from conventional pathology approaches, immunostains, et cetera, moving to DNA RNA extraction and generating next generation sequencing libraries that then could be taken through different types of data analysis, variant annotation, and interpreting and reporting. And as I mentioned at the beginning, if needed, clinical confirmation so that information could be written to the patient's medical record. What I've outlined here are the assays that are being used. Just briefly, we wanted to look broadly rather than in a focused way across the genome at all genes. So we introduced what we call an enhanced exome that compares tumor DNA to normal DNA. And in particular, not only looking at the known protein coding genes, which defines the term exome, but adding in additional probes that tile along each chromosome in the genome so that we could also report out critically important information about focal, arm level, and segmental amplifications and deletions that in many ways define different diagnostic categories for these patients, may define high versus low risk, so prognostic information, and ultimately may help determine their therapeutic path. We also perform high coverage depth, so 250-fold average coverage depth on the tumor and normal DNA, so that when we're looking at patients in a longitudinal setting, a primary versus a recurrent tumor, we can also understand changes in clonality or genetic heterogeneity in that transition, as well as identifying aspects of therapeutic resistance, for example. RNA-seq is also critically important. Here we're performing this coming out of an extraction from the tumor cells, and we're performing multiple analyses, and I'll walk you through these in a subsequent slide. More importantly, in the CNS realm, of course, with the WHO 2021 guidelines, the use of Illumina EPIC 850K methylation arrays coupled with the DKFZ classifier have become part of the standard of care. We also can perform this for sarcomas, although we recognize this is not medically relevant information per se. And then finally, in the case of defining specific gene fusions in a focused way or in areas where the cancer has low tumor content, we can utilize the ArcherDX fusion plex assays that we have to identify driver fusions. And as I've noted here below, with the exception of RNA-seq, these different assays now have CAP/CLIA as well as New York State Department of Health validation in our IgM clinical testing laboratory. So performing lots of tests in a multiplex way really requires high performance computing. And we really had a choice here whether to design a local high performance computing center or whether to go to the cloud. And we decided on cloud computing and spent a lot of time proving HIPAA compliance and privacy in this Amazon Web Services-based cloud. But this is now the sole source of our computing and infrastructure at IgM. You can see the different assays that I mentioned from next generation sequencing shown here on the top panel. Tumor normal enhanced exome sequencing goes through our Churchill aligner to the most recent genome reference, and then gets evaluated through different pipelines to identify germline, somatic variants of all types. And as I mentioned already, the high depth can give us information about clonality or heterogeneity. Along the bottom, tumor RNA-seq also goes through Churchill alignment to look at different aspects of fusions that can be detected, as well as more recently internal tandem duplications, another form of alteration that can cause a driver within a tyrosine kinase gene. And then we also do differential analysis for expression using a variety of tools to look for druggable outliers, to look for different aspects of immune infiltration, including estimate and deconvolution approaches that I'll talk about briefly, And we can also predict neoantigens from the tumor that look like they are producing specific peptides that may be stimulatory to the immune system in a vaccine setting or otherwise. So just briefly, I think I really don't want to spend a lot of time on specifics, but just to illustrate some of the capabilities coming out of RNA-seq. You can see on the flow diagram to the left of this slide, we're also heavily utilizing the treehouse cohort that's housed at UC San Diego, and we've contributed a lot of data to this cohort and database as well. But these data are used to really help position new patients coming in through our cancer protocol relative to a wealth of data from other types of cancer diagnoses. And this can be critically important, for example, in identifying a patient that doesn't have a clear determinate diagnosis by pathology. Just one aspect is matching their RNA expression profile to those of other patients. And in addition, we also, as I mentioned, look for gene expression outlier analysis. Here's just an example where we're comparing a single patient in the red dot, which is a patient diagnosed with Ewing sarcoma, to a variety of different cancers for the expression of a gene JAK1. And you can see how this is grouping in the treehouse cohort relative to other Ewing sarcoma, and then in our own cohort, relative to a group that includes Ewing sarcoma patients, but basically all non-CNS patients, showing quite high expression of JAK1 in this particular case. We also developed our own gene fusion detection pipeline coming out of RNA sequencing. This is published, so I won't spend a lot of time on it. Suffice to say that in the blue boxes on this diagram on the right-hand side are a variety of different fusion and internal tandem duplication calling algorithms. And so one question you might ask is, why so many? And the answer is, of course, that they all have their own strengths and weaknesses, including false discovery rates that we want to normalize for. And so the approach is shown here in the diagram is we do run the same data set through all of these algorithms in our cloud environment, but we then perform an overlapped evaluation to sort of understand in aggregate what are the fusions that are commonly called across the different algorithms. This can allow us to prioritize those fusions, and then we go through a series of knowledge-based filtering steps, including a step that may bring back into consideration fusions that have low levels of expression. A great example here is the KIA-BRAF fusion that's commonly seen in pilocytic astrocytomas and other low-grade brain cancers, but is very, very lowly expressed. And so, as I mentioned before, if need be, we can also clinically confirm these fusions and write them into the patient's medical record. Similarly, for internal tandem duplications, that really has been a valuable component of our new identification of drivers that I think was lacking in the past. So here's just one example from a patient with an FGFR1 in-frame ITD. 9 amino acids within the tyrosine kinase domain have been duplicated. And as you can see on the right-hand display through differential gene expression analysis, this patient is reporting an outlier expression level for FGFR1, which is coincident with this driver alteration, gain of function. So here's a nod towards the Cicero algorithm. It comes from the group at St. Jude. You can see the PMID for the original publication at the bottom of this slide. And this has really been transformative in terms of our ability to identify these ITDs. Here's just a few examples from some of our patients in terms of different ITDs that have been detected. And these, in most cases, have been clinically confirmed, as well as in some cases, even evaluated on long-read sequencing technology, such as the Pacific Biosciences instrument, as illustrated on the far right-hand side of this table. So you can see EGFR, FGFR1, and one patient with a PDGFR beta, as well as a FLT3. And this continues on because many of these are, of course, targetable alterations as well. Finally, from the standpoint of immune infiltration, which is of great interest to me personally in my own research program and to others as well in the pediatric setting, and it sounds like you'll be hearing from this about this from Trevor Pugh in the next weeks. We do use a variety of different approaches, including cybersort-based deconvolution shown here, to really evaluate whether immune infiltration has become a part of the tumor microenvironment. And we especially like to study this in patients with longitudinal specimens, so multiple recurrences, for example, that have taken place over the lifespan of the tumor. Our interest in immune infiltration also extends to patient samples that may not perform so well because they come from FFPE and the RNA is quite degraded. In those cases, we substitute in the nanostring immunoprofiler assay. This is just a brief view of the technology here from nanostring if you're not familiar with it. Really a platform that specializes in being able to deal with highly degraded RNA samples using a series of gene-specific barcodes that are the equivalent of a fixed content assay. About 730 different genes can be evaluated here, and then essentially sorted into different immune response categories that characterize different types of immune cells, as you can see from this graphic, and tell us more information about chemokine expression, for example, interleukins and so on. So this can be a surrogate in place of deconvolution from bulk RNA-seq. And in my time at WashU, and since then, I've also been very pleased to be a part of the group that has developed out PVAC tools, which was recently published in Cancer Immunology Research, and is a fully open source set of tools that can be identified and downloaded from the pvac.tools.org URL at the bottom right on this slide. This is just a series of different tools that link together and really allow you, without advanced bioinformatics expertise, to take a series of mutations and turn them into evaluated neopeptides in the context of a patient's HLA haplotypes for type 1 and type 2 neoantigen prediction. So a variety of functionalities here. Again, this is published, and I would welcome you to take a look at PVAC tools if that's something that you're interested in. So to wrap up this part of the talk, I just want to show sort of our progress to date. These are current numbers, as you can see, from the end of July this year, where we have different metrics around the sex of patients that have been sequenced, patient age at diagnosis. So you can see in some cases we are studying patients that are in the category of adolescent young adult or AYA patients, as well as pediatric patients down to less than 1 year of age. And in the far right pie graph, the different types of malignancies that have been evaluated, including the 46% of which are CNS malignancies. So we've had a significant uptake on this protocol over time by our neuro-oncology and neurosurgery providers, as well as others, obviously. And for those of you who are interested in the subset of cancer gene-driven abnormalities, you'll see the 17% of this protocol that has actually evaluated children in our vascular malformation and overgrowth syndromes clinics, often identifying PIK3CA and other alterations that are somatic mosaic in these tissues that are affected by malformations or overgrowths. This just shows you the growth of the protocol over time, if you will, from when we launched in 2018 to the most recent data. And the numbers in the red box correspond to the increase from between 2018 and 2021. Now, as I mentioned, as of this year, we did take these assays to orderable clinical tests. So we do appreciate that much of the research protocol devoted to multiplex characterization of our cancer patients over time, now that we have clinical orderable assays from our CLIA lab, will decrease just as a natural progression from research to the clinic. And of course, that was the goal all along. Finally, I'll just finish up with a graphic here that shows you the aggregate of patients accepted onto the protocol. At the time this was made, end of July, 450 patients were evaluated by our study team. There were a proportion of these that were not consented. You can see the breakout of reasons on the far left for lack of consent. And then of the consented 400, a subset that weren't sequenced, the majority of which are sequencing that's still in progress. But there were some samples that were not received, uninvolved with disease, or had insufficient tissue for our assays. Importantly, along the bottom, you can see the aggregate yield, if you will, of medically meaningful findings, which we define as diagnostic, prognostic, or therapeutically relevant to the patient. And you can see that spans from germline cancer predisposition, fusions, potentially targetable expression, medically meaningful copy number, and medically meaningful somatic variation. For those of you who may wonder, the numbers on the people shapes on the left of each of these boxes are the number of patients where this type of finding was made. And on the DNA double helix were the number of variants in total that were identified in that number of patients. So there's not always a one-to-one correspondence here. Well, in fact, there's rarely that other than maybe somatic fusions. So I want to just briefly take a couple of minutes to illustrate some cases from this study and just try to bring together all of the tools and technologies that I've described for you to illustrate what it is that we've been doing, again, in a couple of cases and in the setting of precision oncology. So our first case comes from a patient with an atypical neuroepithelial tumor. And this patient really falls into the category of what we commonly refer to in about 10 to 15% of CNS malignancies as indeterminate diagnosis. So the pathology coming back from this patient, which, as you can see from the bullet points on the left, has recurred on multiple occasions: a primary disease diagnosis in 2015, recurrence 2 years later, and a second recurrence in 2020, all of which were gross totally resected. The pathology diagnosis varied a little bit from time to time, but essentially was in aggregate indeterminate. And we were able to study initially the primary tumor from 2015 from formalin-fixed material, and the two recurrent tumors in 2017 and 2020, both from fresh frozen. With the type of RNA analysis that I described in the fusion prediction pipeline that I walked you through, we identified at the time a novel EP300B core fusion that was confirmed clinically, present in all three tumor samples, and corresponded to B core IHC as an activating fusion. You can see in the pathology IHC image on the lower right. But in 2018, when we initially evaluated this patient, just looking at the 2015 and 2017 tumors, both of which contained this EP300B core fusion, there were no reports of these fusions in the literature. And so the most that we could equate this to was some TCGA data that had identified a B core ITD. DNA methylation analysis of this patient's sample showed that there was not a reportable score of concordance. This is from an older version of the classifier. The newest version of the classifier, version 12.5, is shown here, and this was rerun. And this, of course, now because of additional reports of B core and B core L1 fusions in the literature, has shaped out a new methylation class in the version 12.5. And shows that this patient actually still doesn't have a reportable score, but at least is grouping closely with these tumors. So a reportable score for us is above a 0.85 confidence score. This is at 0.22, as you can see. The new evidence supporting the EP300B core fusion driver came along in 2019 in this paper that reported 3 additional patients with pediatric gliomas, distinct clinical pathologic features, and as you can see from this figure from the paper, a variety of different B core EP300 fusions, one of which matched to the child that we had identified as well. So this was important and valuable new information coming from the literature. And as reported in that 2019 paper, and also as illustrated here for our patient, EGFR is overexpressed as a consequence of the B-core, EP300 B-core fusion. So you can see some dots here in red pointing to the different time point samples from our patient that I described earlier, the highest expression of which was in the 2015 sample. And this is probably for reasons that will become apparent as I show you some of the subsequent data here in the next few slides. So what we identified in this patient in the longitudinal evaluation, here just shown for the 2015 primary and the 2017 recurrence, is that on the left, we see evaluations of the RNA-seq from estimate, which is a published algorithm illustrating that the estimate immune score, second down from the top under stromal, goes from a negative value to a very highly positive value. We also were able to study this patient using the nanostring immunoprofiler assay, and you can see the difference between recurrent and primary. Primary is on the bottom where everything's fairly blue or black, unchanged, compared to the recurrent, which scores highly relative to a number of other patients in the consequence of being identified with increased immune infiltration. In particular, this patient is sort of across the board, has lots of infiltration, but is showing significant amounts of infiltration in CD8 T cells, cytotoxic cells, and other immune cell types. So we went back to pathology colleagues and asked for some additional immunohistochemistry stains to try and substantiate these results. They're shown here for the recurrent tumor at the top, the primary is at the bottom, and for a variety of different stains. So CD45, generally any heme-derived products such as immune cells writ large, and importantly, on the left-hand side, macrophage monocyte markers that illustrate increased infiltration in this setting. Finally, we looked at the changes in the tumor neoantigens over time, comparing the primary with the recurrent sample. On the left, we're just looking here at primary and recurrent variants. We can see these are related tumors. So they share variants such as the JADE2 and PM20D2 that are in the center of this graph. But obviously, the recurrent tumor graft on the y-axis has a larger number of variants and ones that are highly unique to it, just in terms of genes with non-synonymous mutations, that is. When we take that information, compute peptides from those, and then run them through PVAC-seq, what we can see shown on the right-hand side is that primary tumor compared to recurrent has far fewer predicted neoantigens. So this increased neoantigen burden may correlate with the infiltration of T cells in particular that may be responding to those novel peptides. Just one other quick example in a pediatric CNS cancer. This is a case of Lynch syndrome associated glioblastoma. You can see the patient's images here showing a recurrence in a very, very short time frame following an initial analysis or diagnosis by imaging on the far left. A gross total resection is illustrated by imaging, and then just a month later, evidence of significant recurrence. This patient then received a couple of rounds of radiation therapy with veliparib followed by temozolomide. But upon follow-up in January, just the next year, clearly had not responded to these therapies and needed another gross total resection. It was at this point in time that we studied this patient on the cancer protocol, and you can see that this is a teenager. So what we identified in this teenager is Lynch syndrome. So in the germline, a pathogenic founder variant at about 50% variant allele fraction. So characteristic of a cancer predisposition. And then the corresponding somatic variant in PMS2 is illustrated below in the second table, along with polymerase epsilon, other Lynch syndrome genes, really acquiring many, many mutations as a result of this insufficiency of PMS2 writ large. And in aggregate, I think over 15,000 mutations in the patient's exome data. Upon methylation analysis, we get close to a reportable score, but not quite. And this is, again, the 11.5 version of the methylation profiler. I think there are increasing number of these patients now represented in the version 12.5, but again, an inability to classify at this point in time. And predictably, this patient, because of the high mutation load that I mentioned, because of the Lynch syndrome diagnosis, which was clinically confirmed, was assigned to nivolumab therapy and remains to this day, the slide's a little out of date, but remains to this day with no evidence of disease, responding nicely to continued nivolumab therapy. And I know there are other instances like this in this patient population. So one example where immune checkpoint blockade therapy can play a role in the pediatric AYA setting. If we move to the evaluation of immune infiltration, what we can see on this slide is results in the center and the colorful graph from CyberSort deconvolution, looking on the bottom at the primary tumor and then 3 different regions from that recurrent resection just a few months later, each with different tumor cellularity, which corresponds to the size of the bars, but all with significant information around significant macrophage and monocyte infiltration in the recurrent setting. This is supported by the immunohistochemistry stains at the far left with CD163, a macrophage marker. So the aggregate experience here in looking at longitudinal CNS malignancies was one of the aspects of data that fueled a successful competition for a U54 grant that we received a couple of years ago now in the Pediatric Immunotherapy Drug Development Network, part of the Cancer Moonshot. And this is really allowing us in the preclinical setting to evaluate different immunotherapy approaches that are specific for pediatric malignancies like CNS and sarcoma. This includes different projects that are focusing on engineered natural killer cells, oncolytic virus-based therapies, and targeting myeloid cell-based immunosuppression, much like the 2 examples that I just showed you. And also my role in this is providing, along with my team, comprehensive immunogenomics evaluations, including some newer technologies that I don't have time to talk about today, such as mass cytometry, also called CyTOF, and single cell RNA sequencing. So I just want to finish up my remarks and leave time for questions, but I know that people are perhaps aware of the NCI contract that we were awarded mid last year and has continued to develop over time towards multiplex cancer molecular profiling in a much larger population than we could ever garner here in one pediatric hospital. So this is an NCI contract we were awarded. Our initial work so far that I'll talk about today has been with the Children's Oncology Group. And really, this is a revolutionary new approach that includes our Amazon Web Services cloud in a couple of ways that are illustrated here by this diagram. One in particular is that as we bring in samples from COG and our biospecimen repository across the street from us here at Nationwide Children's Hospital that are performing all the DNA and RNA extractions from these tissues and bloods, we're no longer accepting sheafs of paper with a pathology diagnosis and other clinical information about the patient. Instead, these are all being brought in electronically. And so there is a significantly reduced chance for error here that is linking in our LabVantage LIMS system the patient samples with their corresponding paperwork. This laboratory information system also provides the sort of framework or infrastructure that oversees the lab operations, but also the bioinformatic processing, analysis, interpretation, and actual evaluation of data and sign out of the clinical report. So everything is being done electronically. The information is being captured electronically so it can be used future forward to help diagnose and report out on other patients. I'll talk about that in just a moment. But the other important component here from Amazon Web Services is the data delivery piece. So no more faxing clinical reports. Rather, once these are completed in LabVantage, they are grabbed by Amazon Web Services in our HIPAA compliant cloud and served out to the institution and the provider that originally enrolled the child onto the COG protocol. So this secure sign-on portal allows the provider to securely sign on, download the electronic result, and incorporate it, if need be, into the patient's medical record according to their EMR. But I think also eliminates a lot of again, use of fax machines to deliver this information in a very, very rapid turnaround. The last piece of this that I'm super excited by, since I sort of grew up in the Human Genome Project and have been a staunch defender of data sharing ever since then, is that we aren't just keeping this information in-house. All of the information coming out of the MCI, as it's called, is being deposited again through Amazon Web Services into the Childhood Cancer Database Initiative Public Data Repository, the CCDI. And at the bottom of the slide, it says within 90 days. In actual fact, this takes place literally within seconds after the signed out clinical report goes live and is provided out to the hospital and provider that nominated or enrolled the patient onto the protocol. So, you know, these data will be publicly available. They will be available for data mining and download to appropriately approved individuals through the normal process that's utilized by the government in dbGaP to ensure data privacy in the future. And so I think we're not just serving the patient today, but we're serving patients into the future with this wealth of information that will be going into the CCDI. I won't go through this in detail, but this is just now the expanded Amazon Web Services pipeline for the different MCI assays and analytics that are being utilized. So here is outlined from the top, the enhanced whole exome sequencing that I already talked you through, the Archer DX pan-solid fusion plex for the identification of fusions for these patients, and ITDs as well can be evaluated by Archer. And then the Illumina EPIC methyl array evaluation for CNS malignancies that leads to disease classification. We have this validated now for the classifier 11.7, and we're working towards validating that 12.5 version of the classifier that I mentioned. Importantly, and in some cases really remarkably, these clinical reports are being delivered back as signed out reports to providers within a 14-day turnaround time from receipt of the DNA and RNA from the BPC. So their pipeline takes about 5 days. Ours takes about 14 days in total for all 3 assays to be processed, analyzed, and signed out by our clinical directors. And this delivers information in a clinically relevant time frame for these patients. I won't say anything about this slide other than to just allow you to gaze upon the inner workings of what was required for all of our bioinformatics to implement that beautiful purple diagram that I showed you a couple of slides ago, and just to really be amazed, as I always am, that we were able to bring this concept to plan in the computational environment that includes our clinical informatics in Amazon, our LIMS and LabVantage, the cloud solutions team that allowed for all of the data intake and clinical report and CCDI deposition of data, and the translational bioinformatics team that really put all of the pipelines together for us to compute on these data. And then finally, I'll just show you a snapshot of where we're at with the MCI clinical testing. This is now open to CNS and sarcomas, soft tissue sarcomas in particular. Next Monday, September 12th, it will go live for rare cancers. And so we'll see another uptick of cases. But right now, today, we've looked at about 250 patients. In aggregate, 743 orders have resulted from those patient samples. You can see the number of orders by test on the right, upper right. Obviously, soft tissue sarcomas, or STS, only recently began accruing into this protocol. CNS was first, so the numbers are larger there by logic. And then you can see along the bottom the numbers of different tests that have been completed for the specific assays: methylation, fusion in the center, and then tumor normal exome on the far right. So this is current as of 8/29. We are excited about the progress here and anxious to see how this plays out over the next several years of the project itself. One of the, and I'm just finishing with this, so we'll have time for questions, but one of the challenges, of course, is scalability. We're already anticipating this challenge in a number of ways. I just wanted to show this diagram and would focus your attention perhaps here in the center of it on Barhouse, which is essentially the database where we're taking in lots of information from the genomic testing that's being performed here, but also importantly, constantly surveying the Internet of Things for new information that might be pertinent to the clinical studies of these patients, and bringing that information in for future consideration as we see results being signed out on new patients that have already been seen in other patients, but new information about those particular alterations in the genome that may be pertinent to diagnosis, prognosis, or therapeutic assignment. So I think this is sort of the wave of the future. This includes aspects on the final bullet point here of machine learning approaches that will allow us to do multi-omic data integration for diagnosis. So looking not in separate windows at germline, somatic, fusions, and methylation, but being able to aggregate those together to really understand if they make sense in the context of what's already known, or if indeed they are identifying new alterations within disease types that have not been previously seen. So just to finish then, for conclusions and next steps, I think hopefully you've heard enough about the clinical genomic assays of pediatric cancers, adding important clinical information, treatment planning, diagnosis, prognosis. I'm committed to combining DNA and RNA results. I think this is an incredibly powerful combination and tells us much of what we need to know. I'm sure there are other components that could be added as well, but these are tractable within the time frame that we have available to us. I didn't talk about it today, but we have really, through the Translational Cancer Protocol, enabled new hypothesis-driven research, and certainly the U54 that I mentioned is one aspect of that. And then lastly, scaling the molecular profiling is going to be our next focus. And I've shown you an example of how we hope to do that with automated variant interpretation using machine learning. So I'll finish by just acknowledging the group. It does take a village. Everyone has been phenomenal in this, and I especially want to acknowledge our collaborators in the clinic, specifically in the cancer, oncology, pathology, and surgery areas of the hospital, as well as my collaborators on the U54. And of course, always special thanks to patients and families for their courage to participate in our studies and hopefully to benefit from them over time. So that's what I had prepared today, and I'm happy to address any questions. I think Jay was going to assist with those. Thanks.
Thank you very much. That was a great lecture, very informative, and it's an exciting time for what I would say is it's an exciting time for families that have a kid diagnosed. Could be a real game changer. I had one quick question before I jump into the questions that were submitted. In your flow diagram back there, GC, I guess, is genetic counselor.
Yes.
So you had a separate path for genetic counselor and clinician. Sometimes they would work together, I would imagine also, right?
Oh, absolutely. And like most pediatric hospitals, we have a specific clinic where oncologists in particular and genetic counselors come together in the setting of a germline finding to really counsel the patient and family, talk about some expanded testing and really what this means for the family and that sort of thing. So absolutely. And, the other area where genetic counselors for us are critically important is in consenting patients and families onto the protocol, helping them to understand what we're going to be doing, what kind of data there is, what's going to be collected, and how it will be shared, and of course getting their approval or non-consent as appropriate on all of those different areas. So it's a very specialized art form there.
I thought it was also interesting on the diagram. It said if you come back with a negative to a diagnosis, you run the sample again.
We do. Typically, we keep the data because the samples are hard to come by. So we keep the data, and this is really some of what would be triggered by that new knowledge coming in from additional reports in the literature or new FDA-approved drugs, et cetera, is that we do want to have that capability to revisit over time that information, especially when it may be critically important with a recurrent tumor or that sort of thing. So yeah, that's part of the plan.
All right, so jumping into the questions that were submitted, the first one said, what percentage of patients in genomic tests done were covered by insurance?
Oh yeah, that question. So so for the research protocol that I talked about, the translational protocol, just to be very clear, the cost of that testing, analysis, et cetera, was all covered by the Institute for Genomic Medicine. Now that we've moved into the clinical realm, of course, this becomes critically important, and certain assays obviously are better covered than others. So fusion analysis is almost always readily covered by insurance companies and CMS. Methylation only in the CNS setting is getting reasonably well reimbursed because it's part of diagnosis, and that's understood because it's in the WHO guidelines. The big challenge is the tumor normal sequencing. And, we are attempting to get insurance reimbursement for these patients in this type of testing. In many cases, that is not possible. And so that is being covered by the hospital just because they firmly believe in the work that we're doing here and the fact that it's going to be important and transformational for patients and families. I think over time, we will try to build engagement around the importance of reimbursing the tumor normal exome. But, I think that's something that we'll need to have a significant amount of information available to us, especially in that area of how this impacted the patient's prognosis, diagnosis, and treatment plan, and what the outcomes of those treatment plans were. So it's a big package that has to roll together over time.
So the next question says, this is really great work, and I'm excited to see what the future holds when this approach is applied on scale to kids with cancer.
Yeah.
How far off do you think this precision approach is from being applied widely at diagnosis to determine clinical treatment?
I mean, my hope is that within this 5-year time frame, we're really able to show the scalability and the applicability. I do think that there are significant barriers. So one of the ones that I'm looking at my acknowledgement slide is that we've brought on additional clinical lab directors as well as variant interpretation scientists. But at the end of the day, even with automation, we may need to figure out a clever, probably AWS-based approach to engage other clinical lab directors, variant scientists, and/or molecular pathologists to help in the sign out of these, these tests. And so I can imagine a distributed network, if you will, of qualified experts in the clinical setting that could access these test results through the AWS, evaluate the data as they would in any other case, and then sign out that result. Ultimately, I think I would love to see this move to a nationwide, no pun intended, scale where every child, even if they were diagnosed at a rural hospital, could have the availability and access to this type of testing. But I do feel like this pilot or this proof of principle that we're working on now really has to be a success in order for us to take it to that next level.
So what you're saying is it could be scaled, but the infrastructure would have to be spread out.
Yes, I think the infrastructure and the expertise for clinical sign out.
All right. The last question here said, has there been follow-up based on the profiling with targeted therapy? For instance, with the high immune infiltration CD8-HI, has there been follow-up with immune therapy? Do you see a difference between tumor types and the correlation between immune infiltration and treatment response?
Yeah, great question. Obviously didn't have time to talk about it today, but we do see correlation in these longitudinal samples. In particular, in CNS malignancies, where I've focused much of my research, which I didn't have time to talk about today, and that wasn't the focus for the talk. High-grade malignancies do seem to have a characteristic immune infiltration in the recurrent setting. That said, recurrences are more characteristic of high-grade malignancies. Low-grade, we have seen recurrent tumors. It's rare, but we have studied those. And in general, we find much less evidence for immune infiltration in the low-grade disease in the recurrent setting. So this seems to be an aspect of high-grade disease that tracks with adult studies that show the same thing in gliomas, glioblastoma in particular, which is most frequently studied. So we think that there are aspects to immune suppression that may be either encouraging the recurrence to grow, or in the course of recurrence, setting in an aspect of immune suppression that many of the studies in that U54 that I mentioned are really focused on alleviating the immune suppression and allowing therapies to work there in the setting of immunotherapy. So we have a ways to go, but I think we're seeing good concordance with our animal models and some early results from the oncolytic viral therapy approach in particular, as well as the natural killer cells that are indicative that we're on the right track, at least in the preclinical setting.
And do you see the same between brain and spinal with high grade and low grade?
We do, although the number of drop metastases that we've had to study is precious few. Certainly are quite interested in that, but the small numbers, probably on the order of 5 or 6 patients that we've looked at there, do show that even if it's a drop met, you will see immune infiltration as well. We're currently working on ways also using that nanostring immunoprofiler that I mentioned, which now has the low input capability. We can evaluate RNA isolated from CSF or plasma to also look for evidence of recruitment of myeloid cells from the bone marrow compartment into the tumor site. So this is part of some correlative science that we're proposing in a very exciting targeted therapy trial for high-grade CNS malignancies that's going in today.
Oh, wow.
Yeah.
Well, that's the last of our questions.
Okay.
I just want to thank you again for joining us today and sharing this exciting project.